Welcome back to G Fun Facts Online. Today we are diving into a topic that sounds like it was ripped straight from the pages of a science fiction novel, but it is actually happening right now in the laboratories of advanced biotechnology. We are talking about the future of cancer treatment. And no, it is not just about making stronger, more aggressive drugs, it is about something much more sophisticated. It is about smarter delivery. I want you to imagine a microscopic missile, but this missile is not made of metal or circuits, it is made entirely of DNA. And this DNA is folded into a precise three-dimensional shape, carrying a chemotherapy payload directly into a tumor, while leaving the healthy tissue right next to it completely untouched. This technology is called Aptamer Missiles. It is a revolutionary biotechnology that promises to transform how we treat retinoblastoma and other ocular cancers. In this episode, we are going to break down exactly how this works, the science behind the DNA shapes, and the life-changing potential this holds for patients. To understand the brilliance of Aptamer Missiles, we must first understand the enemy they are fighting. We need to talk about retinoblastoma, often abbreviated as RB. This is the most common eye cancer in children, and it typically affects infants and toddlers. It begins in the retina, which is the light-sensing tissue at the very back of the eye. For decades, the way doctors have fought RB has been with what we might call blunt instruments. The traditional approach involves systemic chemotherapy. This is where powerful drugs are injected into the bloodstream. Now, while these drugs do kill cancer cells, they are not selective. They attack healthy cells throughout the body as well. This leads to the side effects we are all familiar with, like hair loss, immune suppression, and potential long-term damage to organs. Another method is intra-arterial chemotherapy, where a catheter is threaded through the groin all the way up to the eye artery to deliver drugs. This is more targeted than systemic chemo, but it is highly invasive and risky, especially for small children. In the most advanced and heartbreaking cases, the only way to save the child's life is enucleation, which is the surgical removal of the eye, resulting in permanent vision loss. The fundamental challenge here has always been the blood-retina barrier, or BRB. The eye is what we call a sanctuary organ. It is biologically designed to keep foreign substances out to protect our vision. But that also means getting drugs in usually requires high doses or invasive needles. Scientists realized we needed a way to slip a Trojan horse past these defenses, one that only attacks the enemy. This is where the aptamer enters the story. You will often hear an aptamer described as a chemical antibody, but technically it is a short strand of synthetic DNA or RNA. Now, usually, when we think of DNA, we think of the long, twisted ladder, the double helix that carries our genetic code. However, single-stranded DNA is flexible. It can fold upon itself, creating loops, hairpins, and complex 3D knots. This is the core concept you need to grasp to understand this technology. It is about the power of DNA shapes. Think of it as a key and lock mechanism. Just as a metal key is cut to a specific shape to open a specific lock, an aptamer is folded into a specific 3D shape that fits perfectly into a target molecule on the surface of a cell. Because this folding is precise down to the atomic level, an aptamer can distinguish between a cancer cell and a healthy cell, based solely on the unique proteins or antigens sticking out of their surfaces. When researchers attach a chemotherapy drug to this guided DNA shape, they create an aptamer drug conjugate, or more coolly, an aptamer missile. So how does this missile actually work? The process of using aptamer missiles to treat eye tumors involves a sophisticated dance of molecular engineering. We can break down the mission profile of one of these microscopic weapons into 4 distinct phases. Phase 1 is target acquisition, where the homing beacon. Retinoblastoma cells are covered in specific proteins that are rare or completely absent on healthy eye cells. 2 of the most common targets utilized by scientists are nucleolin and EPCAM. Nucleolin is a protein usually found inside the nucleus of a cell, but in rapidly dividing cancer cells, it strangely migrates to the surface. EPCAM, or epithelial cell adhesion molecule, is a marker often found on cancer stem cells, which are the ones responsible for tumor growth and recurrence. Scientists synthesize a DNA aptamer that folds into a shape that binds only to nucleolin, or EPCAM. This serves as the missile's guidance system. Phase 2 is loading the payload. The aptamer needs a weapon. The drug of choice is often doxorubicin, a very potent chemotherapy agent. Doxorubicin has a natural affinity for DNA. It likes a process called intercalation, where it tucks itself between the base pairs of the DNA strand. The aptamer is designed with a specific stem structure where doxorubicin molecules can hide. This is the cargo hold. This design protects the drug from degrading and, crucially, prevents it from leaking out and damaging healthy cells while it travels through the body. Phase 3 is infiltration, acting as the Trojan horse. When the aptamer missile is injected near the tumor or into the eye, it floats harmlessly past healthy retinal cells, such as Mueller glial cells, because its shape simply does not match their surface receptors. It cannot dock, so it moves on. However, when it bumps into a retinoblastoma cell, the aptamer clicks into the target protein like nucleolin. This docking tricks the cancer cell. The cell membrane wraps around the aptamer protein complex and swallows it, pulling the missile inside the cell in a process called endocytosis. Finally, phase 4 is detonation. Once inside the acidic environment of the cancer cell's interior, specifically the lysosomes, the DNA shape changes or degrades due to the acidity. The cargo hold opens, and the doxorubicin is released. The chemotherapy then attacks the cancer cell's nucleus, stopping it from dividing and causing the tumor to shrink or die via apoptosis. But the story does not end there. There is an even more advanced version of this technology called DNA origami. If a simple aptamer is a single homing missile, DNA origami is like an aircraft carrier. Scientists use hundreds of DNA strands to weave 3D nanostructures, such as cubes, stars, tetrahedrons or pyramids, and tubes. This allows for something called multivalency. A simple aptamer might have one binding site. A DNA origami tetrahedron can have 4 Aptamers attached to its corners. This makes it bind to the tumor much tighter, like holding on with 4 hands instead of 1. Furthermore, these structures can carry larger payloads. A DNA nanostructure can carry not just chemo, but also gene silencing tools like siRNA or heating agents for thermal therapy, all packed inside a hollow DNA cage that only opens when it finds a tumor. Recent studies have shown that DNA tetrahedrons loaded with doxorubicin and guided by Aptamers can penetrate deep into dense eye tumors better than free drugs, helping to overcome drug resistance. So why does all of this matter for eye cancer patients? The implications are profound. First and foremost is saving vision, or what doctors call globe salvage. Current treatments often damage the delicate retina, leading to vision loss, even if the cancer is cured. Aptamer missiles are so specific that they spare the photoreceptors, the rods and cones that allow us to see. This drastically increases the chances of saving the eye and the sight. Secondly, there is zero systemic toxicity. Because the missile does not release its payload until it is inside the cancer cell, the rest of the body is protected. This means no nausea, no hair loss, and no damage to the developing heart or kidneys, which is a common risk with doxorubicin in children. It also preserves a stronger immune system to help fight the cancer. Thirdly, this technology helps in overcoming drug resistance. Tumors are smart. They often learn to pump chemotherapy drugs back out before they work. However, aptamer missiles enter the cell via a different door, through endocytosis, bypassing these pumps and effectively sneaking the drug behind enemy lines where the tumor cannot expel it. Looking at the road ahead, aptamer missiles are currently the stars of preclinical research, showing incredible success in cell lines and animal models. The next frontier is the path to the hospital. One challenge is stability. DNA is naturally fragile because enzymes in the blood love to eat it. Researchers are currently using chemical armor, essentially modifying the DNA backbone, to make these missiles indestructible until they reach their target. We are on the cusp of seeing these therapies move into human clinical trials. The FDA has already approved Aptamers for other eye conditions like macular degeneration, which paves a regulatory highway for cancer-fighting Aptamers. In conclusion, we are witnessing the dawn of molecular surgery. Aptamer missiles represent a massive shift from carpet-bombing cancer to sniping it. By harnessing the simple, programmable nature of DNA shapes, we can guide chemotherapy into the heart of an eye tumor with unprecedented precision. For the parents of children with retinoblastoma and for patients facing ocular melanoma, this technology offers something more valuable than just a cure. It offers a future where surviving cancer does not mean sacrificing the ability to see the world. Thank you for listening to this deep dive on G Fun Facts Online.